Welcome to week 2. You should be done with the scientific method, and today's lesson you'll be applying the scientific method. So by now you should know what an observation is, the types of data, how to formulate a good hypothesis, and the difference between independent and dependent variables. So just to recap, so an observation, we use our senses to gather information. It involves making measurements and collecting data, and there are two types of data: quantitative, which is numbers, and qualitative, which are descriptions. A hypothesis, again, is not just a prediction. A good hypothesis gives examples and good explanation for the reason why something occurs or why a phenomenon occurs. So this is an example of not a good hypothesis. If we add phosphorus to the soil, plants will grow or will increase. Now, a good hypothesis has an explanation. A good hypothesis sounds like this: if we add phosphorus to the soil, plant growth will increase because phosphorus is essential for growth processes such as energy transfer and photosynthesis. In an experiment, there should be variables. There are independent variables, dependent variables, and control variables. So an independent variable is what is controlled or manipulated by the researcher or scientist, and the dependent variable is what is affected due to that change. And the control variables are what conditions remain the same. Okay, so for this experiment, you'll be growing seeds, preferably mango beans, just because they grow faster, you can see better results. But if you can't find any mango beans, I think any beans would be fine. So you'll be doing your observations for about 5 days. If you need more time, that's fine. So the things you need are containers for your substrates. I have seed starters. So the two substrates that I'll be using are sand and soil. So what you need to do is fill your containers with your substrate. So in this case, my independent variable will be I'll be changing the substrates to see which or where your plants will grow faster in terms of height. And that will be my dependent variable. I'll be observing plant height. So I'll be doing measurements based on height. So your dependent variable, again, is your observation. So you can do number of leaves, you can do plant height for your observations. So I'll be using these two to grow my plants in. Again, and then your control variable are the conditions that remain the same. In this case, the pot size will stay the same, the types of seeds, the number of seeds, the amount of water, and the amount of sunlight they'll be receiving. So the only thing that I changed is the substrates. So plant your seeds, water them daily, and do your observation for about a week. And take notes. Remember, you have to take quantitative data and qualitative data. So quantitative can be your measurements of the height of the seeds if they start to sprout, and then qualitative data can be descriptions like maybe they started to sprout, the leaves started to turn dark green. So again, you're describing the phenomenon. So when you're in the Google Classroom, go to week 2, and you should see the learning objectives, the required reading, which is what you're supposed to read before doing the lab. And there's also a class discussion. So for this activity, you will see an explanation of the lab activity, which is applying the scientific method, which is a seed growing activity. And on the bottom, you see 2 files. So the first one is the checklist in which I'll be using to grade you. So you must have a title, which is 5 points. Identifying your independent variables is another 5 points. Dependent variables is another 5 points. Your constants or control variables, another 5 points. Your hypothesis is worth 5 points. Quantitative data, here you will need to create a table of all the measurements you took and create a graph based on the 2 cups. So in the table you need to use proper measurements. So if you're measuring using centimeters, be consistent when doing your measurements. If you're using inches, use inches. So put your daily measurements, and if you see no growth, then just put 0 centimeters. And then you also need to create another table for qualitative data. Here you would write all your observations, your descriptive observation based on the days you observed your seeds. The analysis, you would summarize your findings based on the data collected, analyze the graph and tell us what your graph means in 5 sentences or more. And finally, your conclusion, here you would explain your experiment altogether. Tell us about your hypothesis. Was it right? Explain. Was it wrong? Explain. And you can also include, if you were to do this in the future, what possible changes you would make. So this totals up to 100 points. Once you are done, you are going to create a lab sheet or lab report, which I created for you using Google Docs. So what you have to do is make a copy of this document. Do not write on this, otherwise all the changes you made will be seen by your classmates. So make a copy and then put it or rename it as using your name. So the lab report has everything that you need to. Now all you gotta do is create your graph. Again, maybe a line graph or a column, a pie chart would not be a good example. A column or a bar chart would be good. So once you are done, you are going to save it and submit it to me or submit it in the classroom for grading. So pretty much that's the activity. So this will go on for about a week. If you need time, please communicate with me. And I hope to see your report and read your findings.